questions on this part? Otherwise, we have only a few slides left. No, not good. Good. Now, the last point that I just wanted to drive home is, well, you can believe me or not, but I think it's better to let sort of evolution speak for itself, I guess. Like how important this type 1 interferon signaling is, and to sort of bring everything together what we've heard today. And for this, I just need to introduce very briefly that if you think about evolutions with RNA viruses, which are way faster in adapting because they don't have as much proofreading as we have with our DNA genomes. So there's essentially constantly an evolutionary arms race between viruses and cells. And this means that, evolutionary pressure always selects for viruses that are being able to evade the immune system of the host, whereas the hosts usually tend to accumulate mutations that are beneficial for them because they can suppress the viruses effectively. This is known collectively as the Red Queen hypothesis, which comes from the Alice in Wonderland book, where essentially what the essential idea behind it is that it's all about running with certain speeds relative to each other, so it doesn't matter how fast you run in an absolute sense, whether this is 2 kilometers per hour or 100, as long as but the important thing is how to compare it to your enemy or nemesis that you're racing actually, and whether you're moving faster or slower than your direct competitor, let's put it like this. So this is the evolutionary arms race driven by virus-host interactions, is known as the Red Queen hypothesis. Now, just to show you that viruses actually use their super small genomes, which is very precious space for many viruses, to actually code for all sorts of antagonists of the interferon system. Now, you should read this graph, and this is already 15 years old or something like this, so it's really just a summary of some of the viruses. Is basically here you have the beginning of the pathway, RIG-I, the kinases, the transcription factors, and then here the receptor, and then the juxtap pathway that Thomas Dekkers talked about yesterday. And all these pink proteins are basically from different viruses and indicate the state in the whole signaling cascade they inhibit. So the main point that I'm trying to drive home here is that clearly many different viruses of many families have all gone through the evolutionary pressure to express currently inhibitors that really inhibit this type 1 interferon induction or signaling, and most of the times multiple aspects of the pathway. And if this were not important for their survival, they would not do it because their genome coding capacity is very precious. So I would actually say that all current mammalian viruses that caused some sort of pathology and have made it through evolution, they have at the moment at least one interferon system antagonist. And I think this tells you something about whether I'm telling you some nonsense or not, about how important this is, because this is the main thing that stands between them and active infection. Now, 2 more slides left. The main reason why I'm telling you this, and just to come back to this whole RIG-I thing, is that influenza virus, which is the one that gives you in German Grippe, which is a seasonal virus that mutates every year over the different hemispheres and keeps coming back. This is one of the viruses that is very heavily recognized by RIG-I in the cell, so it must have a very strong antagonist to inhibit this. And the first evidence came already more than 10 years ago for this, where actually the same group that also discovered that if you express just the 2 CART domains of RIG-I, if you express this in cells, you see the the ubiquitinated forms right here on the plot. So these are non-infected cells, but they express the ubiquitinated form of the 2 CARTs. But if you infect it now with a wild-type influenza virus, this PR8, you will see that the total levels of RIG-I don't go down, so it's not that it's being degraded or so, but the ubiquitination is no longer happening. So this led to the idea that infections with this influenza virus prevent somehow the activation of RIG-I through ubiquitination. And what they have figured out in sort of the years that came afterwards is that influenza actually inhibits the E3 ligase that puts the ubiquitination on top of these CAR domains, which is TRIM25. So this is one side of the story. Now you can of course look at it in the opposite way. So how important is this actually for this virus? And what these guys figured out is that the protein in influenza that is being made in infected cells, called non-structural protein 1 or NS1 in short, it interacts with TRIM25 and prevents basically the dimerization of TRIM25, which is important for its patient. And they figured out very specifically the two amino acids in NS1 that are important for interaction with TRIM25 and no other proteins for as far as they could tell. And they make now a mutant in the NS1 protein in the actual virus that prevents it from interacting and antagonizing TRIM25, and that's basically what is shown, or the effect, the phenotype that this has, shown in this Kaplan-Meier survival curve. So in essence, what you're looking at here is in pink the percentage survival of mice that are infected with the wild-type strain, and you can see that normally these mice are really unhappy and they die between day 5 and 6, 100%. Now, if you look at the mutant here in black, which is underneath this horizontal line, they only mutate 2 amino acids in the viral antagonist that can no longer antagonize TRIM25, and thereby it prevents the inhibition of ubiquitination of RIG-I. And you can see that just by doing that, the mice are totally fine, and these viruses are not very pathogenic at all. So again, this being, I think, one interesting example of how viruses, in this one particular influenza, adapts to antagonizing the different response of hosts. That's basically it. So this is the last slide. So the main point is that influenza A viruses can make an interferon antagonist protein called non-structural protein 1 or NS1. One of the things that it does is it disrupts TRIM25 multimerization and ubiquitination of RIG-I, thereby preventing its activation. So even though it's recognized, the signaling is not happening, and the virus has a replicative advantage because it doesn't trigger a lot of interferon production. Good. That is it from my end. I will ask again if there's any questions. Anybody that's physically here, you can also come after we stop recording. I will just give it a minute to see if there's anything in chat.